am Daniele Vigo. I am an assistant director at Cambrex Milan and specializing in the development of active ingredients for generic pharmaceutical products. Well, the challenges that we need to overcome when engineering an originator process are mainly related to avoid the process conditions or reagents that could have negative impact on productivity and on efficiency. For example, high solvent volumes and lengthy purifications. In a recent case, for example, we managed to develop circulation conditions for a process to obtain a product that was of high quality and did not need additional downstream purifications. And this is now the subject of a patent application by Cambrex. To improve yield and quality during the development of a chemical process, we make use of different tools, such as multiple automated reactors equipped with probes that afford relevant process data. In most cases, the experiments are planned according to design of experiments, and this way we manage to achieve process models and the knowledge that we need with a limited number of experiments. To achieve the requested bioequivalence for generic APIs, we need to know very well the properties of the original product that is already on the market in terms of particle size distribution, purity profile, and polymorphic form. To achieve this, we work in close collaboration with our sales department, that is our interface with the customers, and we also analyze the existing patent and regulatory documentation.